Hi, my name is Keyla Badillo, and I study biotechnology in the University of Puerto Rico at Maya West. This summer, I've been doing research in the Chemical Engineering Department of the University of Michigan with Professor Lola Eniola. My research consists on staining neutrophils with different antibodies to see if I can uncover some ways in which marijuana affects these types of cells. So in my lab, we have a flow chamber that imitates the blood flow in our vessels. And when you perfuse neutrophils over an endothelial layer, you can see that they adhere to the endothelial layer and they go under it, which is called transmigration. And this is what neutrophils usually do in our blood vessels. They go through them when there's inflammation or infection. However, in drug-treated cells, there's almost no adhesion and no transmigration through the endothelial layer. So it is evident that marijuana and other cannabinoid agonists affect neutrophils in a certain way. So for my experiments, the first thing that I did was try to see if there are CB2 receptors in neutrophils, which are cannabinoid receptors present in other types of white blood cells. For this, I drew blood from human donors and I isolated the neutrophils and then stained them with a CB2 antibody on eyes. This antibody contains a fluorescent dye so that then I could read the fluorescence of the neutrophils using flow cytometry and quantify the amount of antibody molecules that had bound to the neutrophils. As a control, I used the same isotype that the antibody contains, so that if the isotype also binds to the neutrophils, then it could mean that the interaction of the antibody with the neutrophils is non-specific. You can see the results. So I actually got higher density with the isotype. It bound more to the neutrophils than the actual antibody. I ran experiments using drug-treated cells and normal cells, and also activated and not activated neutrophils. So we used FMLP for activation, but the results were similar each time. So it seems like there is no presence of CB2 receptors in neutrophils, and then it could be that marijuana is affecting them through some other molecule or in some other way. So after that, I also stained neutrophils with other antibodies to see what the effect is or what the behavior is of other molecules that are present in neutrophils, such as cell adhesion molecules. So I stained for MAC1, LFA1, and cellulose X, and these are the results that I obtained. So these results are similar to the ones obtained with normal cells. These cells are treated with agonist or with drug. And the only difference is that there's a significant reduction of cellulose X production when cells are activated. So the amount is more than halved. There is a reduction of cellulose X production in activated cells in normal cells, but not this much. So my results show that there may not be CB2 receptors on neutrophils, and also that there may be a possible impairment of cellulose X production in drug-treated neutrophils. But these are just preliminary. It opens the door for more research using different types of agonists, antibodies, different conditions, concentrations, etc., so that more reliable conclusions can be obtained. Thank you.